Hello, everyone. Thank you for your coming. I'm here to present this project, Molecular Basis of Response to Sublethal Stresses. So everyone already knows fruit fly cause a lot of damage, from economic damage, phytosanitary problem, and environment issues, and also market access. And for the post-harvest treatment, the non-chemical treatment like cold, heat, and irradiation become more and more preferred. But the current knowledge gap is we know they can kill the fly, but how and why they kill fly. And for the cold and the heat, they kill fly in the same way or different way, we don't know. And also for the diagnostic issues, for the irradiation, we already have the markers. We know a fly already experienced irradiation treatment or not. But for the heat and the cold treatment, we don't have this kind of marker. If we found a fly, it is a survivor from this temperature treatment, or it's just accidentally coming in, never experienced treatment. We don't have this kind of marker. So that's why we started this project. So we want to identify the molecular mechanism of the post-harvest treatment to fruit flies. We want to study what kind of genes or molecular pathways involved different treatment stressors. And these pathway or genes are same or different for different treatment. And also we want to find the markers for heat and cold treatment on the flies. So for the long-term goal, we want to better understand how and if these concurrent stressors can be combined together, integrated together to kill the flies, to make the whole treatment become shorter and more efficiently and cost effectively. And also we want to build methodology and knowledge for other species. So this study, we focused on the fruit fly, but we hope the method and knowledge can be expanded to many other insects. And also hope this knowledge can help us optimize our current treatment and develop new treatment. So in this project, we focus on two flies, madfly and the Q fly, and egg and larva stage, because they are in the fruit. And this is our experiment plan. So first, we got the egg or larva of the flies, and then we give them cold, heat, irradiation. And then we collect the treated fly immediately after treatment, and also collect some flies recovered from the treatment. So we have 3 time points: before treatment, immediately after treatment, and recovered flies. Then we extract their RNA samples from these 3 time points and do the next generation sequencing. So we compare their sequencing results to know what kind of genes expression upregulated, what kind of genes downregulated, to help us find the molecular pathway of molecular gene target genes. So for the heat and cold treatment, we collaborate with Sadi, Peter Crisp, and Kelly Hill do the biopsy. And for the irradiation work, we went to International Atomic Energy Agency that's in Austria, use their gamma cell irradiator system to work on the madfly and the Q-fly, collaborate with Costas, and this is our postdoc, Antonio's work there. So after we finished the treatment and we did collected the RNA samples to finish the next generation sequencing, we do analysis, and this work was done by another postdoc, Kay. And from this, this is just one result of the example for the heat treatment on madfly. So we found 30 genes, and they belong to 5 different groups related to the cell death, apoptosis, defense of response, protein folding, and cellular response to heat. So these 30 genes are involved in the heat treatment to kill the madfly. And we also got the result for the madfly cold treatment, Q-fly heat treatment, and the Q-fly heat treatment. So very interestingly, we found 7 genes relate to the heat shock proteins. And for heat shock proteins, they are already studied in many other organisms. They relate to the response to different stressors, of course, including the temperature stressors. That's why their name comes from. And they're also involved in the protein foldings. So we found for these 7 heat shock proteins in the medfly, Q-fly, heat, cold treatment, immediately after treatment, and a recovered stage, the blue arrow means their expression go down significantly, and the red arrow means they go up significantly. So we found for the heat and the cold, they show very different. And for the medfly and the Q-fly, they also show some difference. So because we got this heat shock protein, we continually want to functional study. We established an RNAi knockdown method. If we knock down this gene from the fly, we want to say they become more tolerant or become more susceptible, or they may directly die. So we choose one example, heat shock protein 27, and we tried at egg stage and the larva stage. For our RNAi method, we found for the larva stage not to work very well. But for the egg stage, we found for the control, just use water. About nearly 80% eggs can develop to pupae. But after we knock down this heat shock protein 27, nearly no egg develop to pupae. And also this ATPase is a lethal gene. If you knock down, the fly will directly die, so the similar result. So that means this heat shock protein is lethal genes. If you knock down, the fly will die. Maybe that's the reason during your heat or cold treatment, knock down these genes and the fly die. We also use the same method, try other heat shock proteins or other candidate genes. So for this project, the intended end users, that should be National Fruit Fly Council and federal government or state government, researchers or research funding bodies, and also industry and growers. So from this project, the message we got is we found that in each fly species, the gene sets differentially regulated in response to heat and cold treatment largely do not overlap. And the individual disinfestation protocols need to be considered for different fruit flies. And the genes induced during the recovery period are candidate molecular markers in diagnostic flies treated by temperature stressors. And also, we established the RNAi method to knock down the genes in the fly. It can be used on other genes in flies. So this is our end user advocate, Kim Jim's perspective for this project. Aim to better understand how different lethal stresses used in post-harvest treatment affect the cellular biology of tephrated fruit fly. Once understood this information, will allow research to focus on optimal combination of stresses to more effectively kill fruit fly. And also in future, the impact of the research should provide more efficient and cost-effective post-harvest treatment to improve market access option for Australian horticulture industry. And at last, I want to thank all the group members from Murdoch University, me and Professor Yonglin Ren, and our 2 postdocs, Kay, currently already got a permanent position in Western Sydney University, and Dr. Antonius, now he got a research entomologist position in International Atomic Energy Agency, and also PhD student, Fahan, and in Saudi, our collaborator, Dr. Kelly Hill, Peter Crisp, and in Western Sydney University, Dr. Alexei Papanikolaou is our bioinformatist and genomic, and also international collaborator, Costas, and the industry advisor, Kim James. Also thank to PBCRC for their funding support and support for the whole project. And for the future studies, we want to engage more with the industry treatment because this project, all the data we got based from the lab. So for the industry, the treatment can be in different fruit, can be at different temperature, and also treat at different times. So all our data need to be engaged and validated from the industry. And also, we in future, we also want to involve the irradiation work and the low oxygen work. Here you can see we only focus on the heat and the cold treatment. And also further functional study all the other candidate genes to find which one can be the marker genes for the heat and the cold treatment, and look for the molecular pathway. So, okay, thank you for your attention and questions.